In this video, we're going to do a quick recap of all the organelles that you met in your research task. So let's start at the centre of our animal cell here with this structure. The nucleus. Okay, so having a think then about our nucleus. Well, it isn't just the largest organelle in the cell, it's the most dense, and it's made up of a number of different parts. So it's surrounded by a double membrane, which is called the nuclear membrane, and that membrane has pores within it. And these nuclear pores are really important because they allow the messenger RNA that's produced inside the nucleus during transcription from the base sequence of DNA. So it's a copy of the base sequence of a gene called the messenger RNA, and this nuclear pore allows that messenger RNA to move out. Okay. We've also got the nucleolus. Now that's got another very important role. The nucleolus, which is found inside the nucleus, is the site of ribosomal RNA synthesis. So that's where the ribosomal RNA is produced. And then within the nucleoplasm, we can see sort of the denser looking patches, and those denser looking patches are the what we call the chromatin, which during cell division you will have seen the chromatin wound up tightly, and at that time it looks like chromosomes, those X-shaped chromosomes you'll have seen in your GCSE studies, but here in this nucleus the cell is not dividing, so the chromatin is unwound, so it's not wound up into chromosomes, and that's made up of DNA and proteins called histones. So at this time it's all unwound in the nucleus like that. So if we have a look now, so obviously this is a diagram of the nucleus, but today we're going to be talking about different kinds of microscope. This is an electron micrograph. Okay, so this is taken by the electron microscope, and that's a type of microscope that uses an electron beam and passes it through a really super thin sample, and we can then observe very small objects in lots of detail. So although the nucleus we can see under the light microscope, the resolution of the light microscope is limited by the wavelength of light. An electron beam has a much shorter wavelength, and so gives us a much greater resolution, which is why when we use an electron microscope we can actually view these internal structures. So we can see the chromatin and we can see the nucleolus within the nucleus, and we can even make out those nuclear pores in the double membrane. Okay, so our next organelle that we're going to look at is the mitochondria. Okay, so the mitochondria is the site of ATP production via aerobic respiration. Now it's really important we say that because some other parts of respiration, the anaerobic bits, actually happen in the cytoplasm of the cell, and there is some ATP produced in that as well. So it's just making sure that we say that it's the site of ATP by aerobic respiration, because then we're being really precise. Now you do need to be able to label your mitochondria up and know about its internal structures. So if you have a look, it does have an outer membrane, but the most interesting bit really is the inner membrane, because that is highly folded, and it's highly folded to form structures called cristae. And the cristae, these folds, actually contain within that membrane an electron transfer chain, which we're going to meet next year. So that's where you've got some proteins, and their job is to transfer electrons as part of that process of making ATP. Then if you go within that inner membrane to the centre of the mitochondria, you can see you've got an area which is called the matrix, and there you can see a little reference to a retro movie called The Matrix. So in the matrix, that is the region where there are some other parts of aerobic respiration taking place called Krebs cycle and the link reaction, and the enzymes involved in those parts of aerobic respiration, Krebs cycle and the link reaction, are found in the matrix. And again, we need to get used to identifying the mitochondria, not just in diagrams like these here on the screen. So these kind of double, we've got here a three-dimensional picture. We've got some cut through two-dimensional pictures as well. We are going to be able to hopefully identify mitochondria in electron micrographs as well. So this is a picture of a mitochondria taken with an electron micrograph. Again, because it's got that high resolution, we can actually see the inner folds of the inner membrane, the crista, and we can even see some rough endoplasmic reticulum alongside here. If you look with these dots, we'll come back to that a little bit later on. Okay, so moving on to the Golgi apparatus or Golgi body. So our Golgi body is a series of flattened discs. Okay, this is important because you can confuse it with the endoplasmic reticulum. The big difference here is if you look at this two-dimensional cut-through picture, these discs are not interconnected, so they're like a stack, like a pile of pancakes if you imagine like that. But things do move from one of these to the next flattened disc, so they do move, and they do that by budding off the end in a little vesicle, and then that vesicle fuses with the next sort of pancake, the next flattened disc, and molecules can move their way through these discs. As they do, these molecules are sorted, modified and packaged. Okay, so an example of the type of molecules this happens to a lot in the cell is proteins. Proteins are produced at the rough endoplasmic reticulum, and then they're carried in a vesicle, and the vesicle arrives at the Golgi body, the Golgi apparatus, and it fuses with that bottom disc, and then as it moves its way through these discs, it will be modified, so it might have some additional chemical groups added to it, like a lot of proteins have. It might be that more than one polypeptide is joined together as it moves through the Golgi body, the Golgi apparatus. So that means that it does get modified and eventually is packaged up in a vesicle. Now this is how two structures that we're going to meet later in the course are produced in specialized cells. So in the epithelial cell, there's something called a chylomicron produced, which is a lipoprotein. You met these high density and low density lipoproteins when you talked about cardiovascular disease at GCSE. So they're produced in epithelial cells. So we will talk about the Golgi body in the epithelial cell, modifying proteins to produce these chylomicrons. And we'll also talk about specialist vesicles called lysosomes. And lysosomes are found in white blood cells, and we'll come back to those a little bit later on. So there are flattened membrane discs of the Golgi body, and you can see one of the key things that will help you identify that you're looking at a Golgi body is the fact that you'll have this stack of discs, but you'll also have lots of vesicles around the outside. And here is an example of an extensive Golgi apparatus, Golgi body, inside a cell taken with an electron microscope. Okay, so you can see that all of these are not connected up to each other, so they're in a stack. It's a very big stack because this is a cell that secretes lots and lots of protein, and because it secretes lots of protein, it's got to package lots of protein up for export from the cell in vesicles, and so that's why this particular cell has such a big Golgi body. But cells only ever have one Golgi body, It's just bigger if they need to secrete more molecules. Okay, so coming now to our specialist vesicle. This is the lysosome. Okay, so the lysosome we do need to know a little bit about. Okay, it's a very large vesicle and it contains a number of enzymes, and those enzymes are called lysozymes. Okay, so there's my lysosome, and inside are what we used to refer to as digestive enzymes. On the new specification, we do need to identify them as lysozymes, so the name of those enzymes. And we're no longer allowed to say that they break or digest things. We have to use the word hydrolyze, so they hydrolyze things. Okay, so here is an example of a worn out mitochondria. So this is damaged, it's no longer functioning. The cell wants to recycle the molecules from which the mitochondria is made. And so what it does is it surrounds that old or damaged mitochondria with a vesicle, and then it uses its lysosomes, fuses them with the big vesicle, releasing the enzymes, the lysozymes, into the inside of the vesicle where they hydrolyze that organelle into its biological molecules, which can be recycled to make something else in the cell. Another thing that cells can use lysosomes for is for sort of autolysis. So this is cell suicide. So if a cell is no longer functioning, it can have within it a series of events initiated so that it releases its own enzymes from the lysosomes. So in other words, it releases those lysozymes into its own cytoplasm and digests itself. So the one thing that we will meet these in is in our immunology topic. So we will meet the phagocytes. They're a specialist type of white blood cell, and they have absolutely loads of lysosomes so that they can do that specialized job of engulfing and hydrolyzing pathogens like bacteria. Okay, so for our next structure, so not really an organelle, but a structure found in the cell. This is one that we'll come back to when we do our mitosis and meiosis topics, because it's involved in the sort of the cytoskeleton, if you like, of the cell, and a particular part of it called the spindle fibres. And you might remember those from GCSE because you'll have drawn them attached to your chromosomes. And you can see it's these centrioles here that at the poles of the cell produce these long protein fibres that attach to our chromosomes and move them around the cell. So for now, just remember centrioles, their job is to be involved in cell division, mitosis, meiosis, and that's through the production of spindle fibres, special proteins. Okay, so that brings us to our endoplasmic reticulum. So endoplasmic reticulum is, as you can see in this beautiful three-dimensional picture, it's a continuous network of interconnected membrane. Okay, so this time the sacs of membrane are interconnected and they're called cisternae. So there's two different types of endoplasmic reticulum, and they're always continuous with the cell membranes. You will find the first type pretty much close to the nucleus. Okay, and that's the rough endoplasmic reticulum. And the reason it is rough is that you can see on its cisternae, it's got lots and lots of tiny little dots, giving it a rough appearance. Those tiny dots are ribosomes, and the ribosomes are the site of protein synthesis. So the proteins are produced on these ribosomes on the surface of the rough endoplasmic reticulum, and then the proteins enter the rough endoplasmic reticulum, and that provides them with a pathway, if you like. So it transports the proteins throughout the cells, or throughout the cell, sorry. The smooth endoplasmic reticulum has a different role. Okay, here is where triglycerides, so lipids, are synthesized in the cell. So if you imagine inside this smooth endoplasmic reticulum, that's where your condensation reactions are going to take place between your glycerol and your fatty acids to make your lipids. Whereas on the ribosomes, the rough endoplasmic reticulum, that's where we're joining together amino acids by condensation reaction to make polypeptide chains. So really, rough endoplasmic reticulum, the ribosome is the site of the protein synthesis, but the rough endoplasmic reticulum transports the protein, and the smooth endoplasmic reticulum is the site of lipid synthesis and transport. Often our examiner will ask you why these sometimes appear to be continuous and sometimes don't. Okay, so depending on the two-dimensional image and where it was sliced through the cell, these endoplasmic reticulum can appear to be continuous, but if they are cut through in a two-dimensional slice where they're not joined, they could look like separate membrane sacs. Okay, so it's just understanding that when you cut a three-dimensional object and look at it in 2 dimensions, then you aren't seeing every plane of view. So in that plane it might not appear to be joined, but in another plane of view it would. So identifying our rough endoplasmic reticulum on diagrams, you're looking for that ribosome stuck to the outside, if you like, covering the outside. Okay. If you're looking at endoplasmic reticulum with an electron microscope, okay, so remember the electron microscope's got a really high resolution, so we can have a very effective magnification, very high effective magnification. So we can look at the internal tiny sort of organelles like the structures like the ribosomes inside cells with this powerful electron microscope. So you can see the rough endoplasmic reticulum stands out because it looks grainy in appearance due to all the ribosomes on its surface. That's going to help you identify that. Again, this is continuous, but doesn't look very continuous in this diagram because, in this electron micrograph, because as we've cut through sliced, if you like, through the sample, and looking at it in this plane of view, we can't see where these discs, these are connected to each other, but they are because it is a system of interconnected membrane sacs. It's just these membrane sacs don't look like they're connected in this particular image. We have a look at the smooth endoplasmic reticulum. That's a lot more difficult. So these are our smooth endoplasmic reticulum here. Now, in terms of an electron micrograph, it's quite difficult to tell the difference between that and a Golgi apparatus. But you can see you haven't got that sort of stack relationship. These are just cut through interconnected membrane sacs. Okay. So having a closer look at our ribosomes now. Okay, so the ribosome is made from ribosomal RNA. So we write that as a little r and then capitals RNA. All right, so we will meet that in more detail when we look at our nucleic acids and we look at RNA and types of RNA later on in the course. We'll also meet our ribosomes again when we look at protein synthesis. So they're going to be really important to us. Okay, in eukaryotic cells, so in the cell types that we're looking at the moment, the plant cell and the animal cell, ribosomes are called 80S ribosomes. So that's a reference to their size. They are made up of a large and a small subunit. And they are the site of protein synthesis. This is where amino acids are joined together by condensation reaction. Now this happens in a process called translation, and that's where the mRNA, the copy of the gene, which is transported to the ribosome from the nucleus where it's produced, is sort of effectively read by the ribosome. In other words, the code in the mRNA is translated to the primary structure, the amino acid sequence of the polypeptide chain that is produced. So depending on the code on the mRNA that is being read, a different primary structure would be assembled, and that is how proteins are, a synthesis is controlled in a cell, and the right mRNA is produced at the right time so the ribosomes can make the right proteins at the right time. Okay, so then we're moving on to the cell membrane, and we already know a lot about a particular molecule in the cell membrane which makes up 90% of the membrane, and that is the phospholipids. So if you remember our phospholipid, it has got a head region made up of glycerol and phosphates, and that head region is hydrophilic and so faces the water, the aquatic environment, whereas the two fatty acid chains, tails, that make up its tails, are hydrophobic and arrange themselves away from water, so forming this bilayer. That makes up 90% of the membrane. But then within that membrane we have got lots of proteins that are arranged in like a patchwork fashion and float around actually within the sea, if you like, of phospholipids. Some of the proteins span the whole of the membrane, like this one here, which is a type of channel protein, allowing some molecules to move through this membrane. Others are just on one side of the membrane and don't span the whole of the membrane. We'll have a very close look at cell membranes in our topic, movement of molecules across membranes. So we know now that they contain proteins and phospholipids. They also contain carbohydrates. Some carbohydrates are attached to proteins to make structures called glycoproteins. They're involved in cell recognition and communication too. So we have lots of different types of proteins with different jobs, like channels, receptors, enzymes that can be embedded in membranes, but all membranes are made up of a large proportion of them are made up by phospholipids. Okay, so something that wasn't in your research task, but I'm adding in now, is something called microvilli. So this is a specialist region of the cell membrane, and the microvilli are finger-like projections or folds of the cell membrane. Now, not all cells have these, but cells that are specialised for absorption, like ones we're going to meet later in the course in our digestion topic, when we look at the epithelial cell of the small intestine, they have specialised adaptations. And in this case, you can see them in two planes of view. So cut through long way, so you can see the long finger-like projections, but also cut through in a transverse section, so as if you were cutting through each of those long folds, and you're looking at the cut ends, so then they appear like little circles. Okay, so both of those are electron micrographs of microvilli, just cut through two different planes. Now, microvilli are to increase the surface area of the cell membrane. This increases the diffusion rate across the membrane because you'd have more space for more channel proteins, more space for more carrier proteins to transport molecules across. Okay, so we're moving now to organelles that are only found in a plant cell. So everything else we've met is present in a plant and an animal cell, but now we're going to concentrate on our plant cell. Now, just like at GCSE, we tend to use the palisade cell as our example, which do contain, of course, chloroplasts, because they are the plant cells found in the leaf of the plant, and so that is where photosynthesis takes place. Please note that if we're looking at different specialised plant cells, then for example a root hair cell, we wouldn't have necessarily all of these organelles. So as they are underground and they don't photosynthesize, you wouldn't expect to find a chloroplast in those. But this is a typical plant cell, as we've looked at a typical animal cell. So zooming in again, but this time just on organelles found in plant cells. Okay, so we're going to start with the chloroplast, which is a really important organelle within the plant, and as I said, in the palisade cells of the leaf cells of a plant. Okay, because it is the site of photosynthesis. Now, you do need to know quite a lot of detail about that process, but we will be teaching you a lot of detail in year 2. So at the moment, we want to just get in the internal structure, the names of the parts of the chloroplast, and also an idea of the kind of reactions that are happening in different places. So it's again a double membraned organelle. And we've got a specialisation to the internal membrane. In this case, it is arranged into discs, which are arranged into stacks called granum. The individual discs are called thylakoid discs, and as you can see, they're green. And they're green because within the membrane of the thylakoid disc, there is the pigment chlorophyll embedded. And the chlorophyll absorbs light energy, and it converts that light energy to chemical energy, which is then used in the stroma, so that's the space inside the chloroplast, to synthesize organic molecules. So in other words, make glucose. So the first set of reactions that happen in the thylakoid, they're called the light dependent reactions because they use light directly. And the second set of reactions are called the light independent reactions because they don't require light directly. Both of those sets of reactions make up photosynthesis. And you can see here we've got an electron micrograph again, a two-dimensional image through the internal structures of our chloroplast, and you would be able to label that up not only as a chloroplast, but you would be able to label up inside the chloroplast the stroma and the thylakoids. In this case, it's pointing at the thylakoid membrane. You can see that these discs are just kind of joined together by a thylakoid membrane, and then the discs themselves you could label up as thylakoid discs, or you could label the stacks as granum in that electron micrograph. By the time you get into year 2 and we've done photosynthesis in more detail, you will be able to describe the light dependent and light independent reactions that happen in both of those parts of the chloroplast. Okay, so moving on to another organelle that is just found in our plant. It's a large central vacuole, which is permanent. So you can get vacuoles in other cells as well, but not large central ones. This contains the cell sap, and it's a way that plants can regulate the amount of water. So if they've got too much water in their cytoplasm, they can put it into the central vacuole. And if they need extra water, they can use the water reserve in there. Okay, but they can also store some other materials in that vacuole as well. So they just help us really to identify plant cells because they're large and central and kind of point in that direction that you're looking at a plant cell. That, and of course, your cell wall, which is, we know, made from cellulose. So this is the plant cell wall. Now, other types of cells, like for example a prokaryotic cell, a bacterial cell, that has a different kind of cell wall. It's made from a different molecule. Equally, the fungi, they have a cell wall as well. So the eukaryotic cells with a cell wall, but the cell wall is made again from something else, a molecule called chitin. So in a plant, the cell wall is made of cellulose, and of course you can tell the examiner these long straight chains are held together by many hydrogen bonds, which makes this a really strong molecule. You can see that it's arranged in sort of scaffolding around the cell. So you can imagine then that your cell wall will provide the cell with strength, okay, so mechanical strength to resist osmotic pressures, but also that plant cell wall still has holes in it, so it's still freely permeable. So water can move in and out of that plant cell with no resistance to it at all. Okay, so that's a really sort of detailed description you should be able to give of the structure of cellulose. Remember, it has the monomer units that are beta glucose, with alternate beta glucose having to flip over. That's why we get these long straight chains. So again, it's linking our two topics together that we've already done. And just reminding you of that key term, when we're talking about lots and lots of cellulose fibres being held together by many hydrogen bonds, we say they form microfibrils. And those microfibrils are what are arranged into larger macrofibrils that make up this matrix arrangement, which is the cell wall. Okay, so that covers all our organelles. Now, one thing we're going to do today is we're going to look at specialised cells, as well as thinking about microscopes. And one of the things you need to do at A level now is to be able to talk about how organelles work together. Okay, so this is a picture of a process that is called protein trafficking, and this is a beautiful way of showing how all the organelles in a cell have got different jobs, but of course these different jobs are enabling them to work together to be that cell type that it is. So here you can see that we've got our messenger RNA, which is a copy of our gene, leaving the nucleus via a nuclear pore. Then you can see it goes to the rough endoplasmic reticulum, and on that little ribosome you can see that it's being used as the code for the order of amino acids that are being put together by condensation reaction to form a polypeptide chain on that ribosome. Then you can see that that little polypeptide chain is moved inside the cisternae of the rough endoplasmic reticulum. And as it's done that, it will fold up into its specific tertiary structure determined by its primary structure. And there it is in its little tertiary structure. And then it buds off in a vesicle. And as it buds off in that vesicle, that is then taken to the Golgi apparatus. And in the Golgi apparatus, this polypeptide chain is sorted, modified, and packaged for export from the cell. And you can see here that we might have a secretory vesicle, so our protein's being secreted, but equally we might be making a lysosome. Here you can see